Yeah, so we are back with another live stream. So in this live stream, we are going to cover the remaining part of the cell engineering. So let's do it. It's small. I will finish it in half an hour. So after half an hour, you can be free. Where was I? Pathology of cell injury has finished. Pathogenesis is also finished. Yeah. Pathogenesis is from pathogenesis I haven't studied. So let's begin. So injury to the normal cell by one or more of the above listed etiological agents may result in a state of a reversible or irreversible cell injury. The underlying alterations in the biochemical systems of cells for reversible and irreversible cell injury by various agents is complex and varied. In general, the following principles apply in pathogenesis of most forms of cell injury by various agents. Okay. Type, duration, and severity of injuries agent. The extent of cellular injury depends upon type, duration, and severity of the stimuli. Example: small dose of chemical toxin cause reversible cell injury, while large dose of same chemical agent cause cell death. Type, status, and adaptability of target cell. The type of cell as regards its susceptibility to injury, its nutritional and metabolic status, and adaptations of the cell to hostile environment determine the extent of cell injury. Example: a skeletal muscle can withstand hypoxic injury for a long time, while cardiac muscle suffers irreversible cell injury after 20 to 30 minutes of persistent ischemia. So, pathogenesis of ischemia and hypoxic injury. Ischemia and hypoxia are the most common forms of cell injury. Although underlying intracellular mechanism and ultrastructural changes involved in reversible and irreversible cell injury by hypoxia and ischemia depending upon extent of hypoxia and type of cell are involved are a continuation of the process. Reversible cell injury, if the ischemia or hypoxia is of short duration, the effects may be reversible or rapid restoration of circulation. Example: in coronary artery occlusion, myocardial contractility, metabolism, and ultrastructure are reversed if the circulation is quickly restored. Okay. So mechanism of cell injury, first is the depletion of ATP, mitochondrial damage, influx of calcium and loss of calcium homeostasis, accumulation of oxygen derived free radical, oxidative stress, defects in membrane permeability, damage to DNA and proteins. So yeah, first the ATP is low. Why? Because the oxygen is not there. Why? Because of the obstruction of the arteries. Okay, so that's why the mitochondria is also damaged. Leakage of pro apoptotic proteins. Okay, then the entry of calcium causes the mitochondrial membrane to disrupt and causes the lysis of the mitochondria, and it also activates the lysosome. Lysosome is a it's a spherical body. It's a spherical body which have lots of enzymes in it. So the calcium triggers the dissolution of the membrane of the lysosome and causes the enzymes to leak out. Okay. Now, reactive oxygen species, these are also very dangerous form of cell injury in which they can disrupt the whole structure of the whole biochemical structure of the cell. Okay. Then membrane damage, when the plasma membrane also damages, lysosomal and membrane also damages. Okay, and protein misfolding and DNA damage, and yeah. Okay. Then, hypoxia is the most common cause of cell injury, which is caused due to ischemia. In ischemia, aerobic respiration as well as glucose availability are both compromised, resulting in more severe and faster side effects of cell injury. Ischemic cell injury also cause accumulation of metabolic waste products in cell. ATP depletion to 5 to 10% of normal cell level will induce cell injury. Due to low oxygen supply to the cell, aerobic respiration by mitochondria fails first. This is followed by a switch to anaerobic glycolytic pathway for the requirement of energy, that is ATP. This results in rapid depletion of glycogen and accumulation of lactic acid, lowering the intracellular pH. Early fall in intracellular pH, that is intracellular lactic acidosis, resulting in clumping of nuclear chromatin. ATP depletion. Decreased generation of cellular ATP causes next processes failure of sodium potassium ATPase pump sodium potassium ATPase, resulting in cell swelling, hydrophic swelling, failure of calcium pump by increased sodium level, where excess of calcium will move into cell, resulting in deposition of phospholipase and cell swelling. Due to hypoxia, ribosome detached from the endoplasmic reticulum and Golgi apparatus, which degrades into monosomes, le- le- leading to decrease in protein synthesis and plasma membrane alteration in the form of bleeding and loss of microvilli, mitochondrial swelling, and small amorphous densities within mitochondria. Okay, this is the thing. Okay, so in this picture we can see the ischemia, mitochondria, decrease oxidative phosphorylation, so decrease ATP. So decrease ATP will give rise to endoplasmic reticulum swelling, cellular swelling, loss of microvilli and blebs. Okay, and less ATP will also give rise to anaerobic glycolysis, low glycogen, high lactic acid, and lower pH. So this um, this um, this whole process leads to the clumping of nucleic chromatin, okay, and detachment of ribosome and decreased protein synthesis. ठीक है. Okay. Failure of calcium pump. Membrane damage causes disturbances in the calcium ion exchange across the cell membrane. Excess of calcium moves into cell, that is calcium influx, particularly in the mitochondria, causing its swelling. Ultrastructural evidence of reversible cell membrane damage is seen in the form of a loss of microvilli, intramembranous particles, and focal projections of the cytoplasm labs. Excess intercellular calcium collects in the mitochondria, disabling its function. Okay. Morphologically, mitochondrial changes are the vehicles in the mitochondria and deposits of amorphous calcium salts in the mitochondrial matrix. Uh, Reduced protein synthesis disperse ribosomes. As a result of continued hypoxia, membranes of endoplasmic reticulum and Golgi apparatus swell up. Ribosomes are detached from the granular endoplasmic reticulum, and polysomes are detached to monosomes, thus dispersing ribosomes in the cytoplasm and inactivating their function. Failure activated phospholipases, membrane damage. Damage to membrane function in general and plasma membrane in particular is the most important event in irreversible cell injury in the ischemia. As a result of a sustained ischemia, there is increased cytosolic inflows of calcium in the cell. Increased calcium activates endogenous phospholipases. This in turn degrade membrane phospholipid progressively, which are the main constituent of the lipid bilayer. So, so activation of intercellular proteases causes cytoskeletal damage. Activity endonuclease cause nuclear damage. Lysosomal hydrolytic enzyme, lysosomal damage, cell death, and phagocytosis. The lysosomal membranes are damaged and result in escape of lysosomal hydrolytic enzymes. These enzymes are activated due to lack of oxygen in the cell and acidic pH. These hydrolytic enzymes include hydrolysis, RNase, DNase, protease, glycosidase, phosphatase, lipase, amylase, cathepsin, etc., which on activation bring about enzymatic dilation of a cellular component and cause cell death. Accumulation of oxygen derived free radicals, oxidative stress. Free radicals are chemical species with a single unpaired electron in an outer orbit. They are unstable and react with other chemicals, cellular proteins and lipids, and initiate reactions in which molecules which converted into other types of free radicals, thereby propagating the chain of damage Fenton reaction. Okay. Free radicals include superoxide, which is converted to hydrogen peroxide spontaneously by the action of enzyme superoxide dismutase. In the presence of metals like iron, hydrogen peroxide is converted to highly reactive hydroxyl radical OH dot by the Fenton reaction. Reactive oxygen species low concentration of the ROS are involved in numerous signaling pathways in cell and thus in many physiological reactions. Therefore, these molecules are produced normally, but to avoid their harmful effects, their intercellular concentration are tightly regulated by healthy cells. The generation of free radical is increased during several circumstances: ultraviolet light, X-rays, ionizing radiations. Reactive oxygen species cause cell injury by three main reactions: lipid peroxidation of membranes, changes in protein and DNA damage. Reversible cell injury, the intercellular changes associated with the reversible injury include plasma membrane alterations like bleeding, blunting, or distortion of microvilli and loosening of intercellular attachments, mitochondrial changes like swelling and the appearance of phospholipid-rich amorphous densities, dilatation of ER with detachment of ribosomes and dissociation of polysomes, and nuclear alteration with clumping of chromatin. The cellular may contain phospholipid masses called myelin figures, which are derived from damaged cell membrane. Copies. So, reversible cell injury has plasma membrane alterations like blebbing, blunting, or distortion of microvilli and loosening of intercellular attachment. Okay. Plasma membrane, myelin figures. Then we have the dilatation of ER with the detachment of ribosomes and dissociation of polysomes. Okay, mitochondrial damages, changes like swelling and the appearance of phospholipid T. Nuclear clumping. Finished? What? Shit, it's finished. Okay, guys. So that was it. Thank you. Bye bye. Love you all. Thank you for joining. Bye bye.